Welcome to this tutorial video. So in today's lesson, we're going to be looking at what we call glycogenesis. Now, you must understand few things before I talk about this topic. So remember, in your foundation biology, you are taught to say the stored form of, energy in human beings is what we call glycogen. When somebody has got excess glucose, what happens to that excess glucose? It will be converted to what we call glycogen. So that's what we'll be looking at today in biochemistry. Glycogenesis. You can look at this word, you just understand this is glyco coming from the word glycogen. Genesis simply means the process of forming it. So glycogenesis is a process that involves the formation of glycogen from glucose. Why are we forming that glycogen? So that we can just have the stored form of energy in human beings. I repeat, glycogenesis is basically a process that involves the formation of glycogen from glucose for energy storage purposes. So this picture that you have here is just trying to show you what I'm trying to talk about. Now, by definition, glycogenesis simply means a biological process in which glycogen is synthesized from glucose for energy storage purposes. So you are forming this glycogen from what? From glucose. That's what we'll be looking at today. Now, you must understand that this glycogenesis process mainly takes place where? In the liver and skeletal muscles. There are other organs that it can take place from, for example, the kidney, but it's at a very minimal level. It mainly takes place in the liver and the skeletal muscles. Specifically where? In the cytoplasm or the cytosol. Now, you must understand that this glycogenesis, we are saying that the liver glycogen is concentrated compared to the skeletal muscle glycogen. What do we mean? You can see here to say the individual liver stores about 100 grams of what? Glycogen. But if you look at the skeletal muscles, remember there are a lot. Your body is surrounded by skeletal muscles. Them, if you now total those different cells, okay, if you put them together, they form about what we call 400 to 500 grams of glycogen. Think of a liver as one bucket, then think of skeletal muscles as different cups, which are a lot. We are saying that liver glycogen is concentrated. Why? It's because it has got just one bucket. Think of it as a bucket which has got a lot of glycogen in it, but you've got a lot of skeletal muscles in your body. They're not as many as the liver cells. I mean, they are more than the liver cells. No wonder if you combine now, you get each specific glycogen amount from each specific cell from those muscles. You put them together, they're going to have a bigger quantity than the liver. But if you talk about concentration, the liver's got a higher concentration of glycogen than each individual muscle cell. That's what we mean. So you must also understand that this glycogenesis process takes place in what we call the fed state. When somebody is eating, that's what we mean the fed state. When we say the fed state, we're referring to a person who is eating. So when you sit at a dining table, you're having your shima or you're having your meal, which has got carbohydrate, sources, you start this process of glycogenesis. Why? It's because as you are eating, your glucose levels tend to increase in number. So I will talk about what we call normal FBS and normal NFBS. So talking about the normal FBS, what I simply mean is that normal fasting blood sugar. When somebody is not eating, what is the normal range of blood glucose levels? So the normal range of blood glucose levels is about 3.9 to 5.5 millimoles of glucose per liter of blood. That's if somebody is not eating, which we call fasting blood sugar levels. Or if you want to put them in milligrams, you can simply say 70 to 99 milligrams per deciliter of blood. So most of you have, I think, done blood sugar level test on the ward. Okay, you get your RBS machine, and then you want to go and check RBS or fasting blood sugar on that particular patient. So what you must understand in this slide is that if you find that the glucose levels in that patient who is not eating is about 3.9 to 5.5, then that is normal. It's within the normal range, because why? We're calling it fasting blood sugar because they're not eating. But if you find that it's above 5.5, let's say it's 6, 7, 8, okay, then that person is in what we call hyperglycemia, which will indicate to you that person is possibly diabetic, because their glucose levels, in as much as they are not eating, they are very high. So you suspect diabetes or hyperglycemia. But if they are low, let's say if the glucose levels are below 3.9, it simply means that they're in what we call hypoglycemia. It's possible that maybe they've stayed hungry for a long time, or they did not eat. So that's about fasting blood sugar levels. Now, what about the normal non-fasting blood sugar levels? Non-fasting blood sugar. So in non-fasting blood sugar, you're talking about the person who is eating, because they're not fasting. And we're calling it non-fasting. So if you are eating, okay, you eat your shima, and then you decide to check your blood glucose levels, they must be at least between 6 to 7.80 millimoles of glucose levels per liter of blood. That's if you are eating or you are in a fed state. Now, you can still diagnose, hyperglycemia or diabetes in a person who is in the fed state. It's because you know that if at all these blood glucose levels they are above 7.8, let's say you've got 9, 10, 11 somewhere there, that is too much. It means you've got excess, blood glucose levels in your blood. Okay? So what happens now is that if your blood glucose levels, we are saying here to say glycogenesis occurs when your blood glucose levels are above 7.80 millimoles. So your body knows how to control normal blood glucose levels. If it sees or it notices to say you've reached 7.8 millimoles of blood glucose when you are eating your shima, it will start now converting that excess glucose to what? To glycogen. Wonder we're saying it occurs in a fed state. But why is it important for you to learn these concepts? It's because when you're talking about diabetic patients, if you're talking about diabetes type 1, they lack what we call insulin. No wonder you give them that artificial insulin so that you can help them, to convert that excess glucose into what we call, glycogen. No wonder here you are seeing that insulin promotes glycogenesis. It's because insulin promotes the conversion or the breakdown of glucose from the blood into something. In this case, glycogen. So that's why you must understand these concepts. Remember, your gluconeogenesis process, when is it going to take place? It's going to take place when you are in a fed state, or when you are eating, or when your blood glucose levels are above 7.8 millimoles per liter of blood. And I've said that insulin promotes glycogenesis. That is even why you give your diabetic patients in the hospital insulin if their blood glucose levels are high, so that you can help them to convert that excess glucose into glycogen. Alright, now just giving a recap here, we are going to look at what we call alpha and beta glucose molecules. So I'm just giving a recap because I don't want to be sounding, as if I'm just mentioning things without giving you background information. So when we say an alpha glucose and a beta glucose, it's very simple. You just look at your glucose molecule on your left here. Alright, this is your first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and the sixth one. But if you come on your first carbon atom here, you find that the OH group on this alpha glucose is down or below the C1 carbon atom. So that makes this an alpha glucose. If you come on your beta glucose on your right here, the same number of carbon atoms from 1, 2, 3, 4, 5, and 6. But if you look at the OH group, it's on top of the C1 carbon atom. So it makes this glucose molecule to become what we call a beta glucose. So I hope you've gotten that difference. Alpha glucose, OH group below the C1 carbon atom. Beta glucose, OH group or hydroxy group above the what? C1 carbon atom. Another recap. You must understand the difference between what we call alpha and beta glycosidic bonds. So when you're talking about an alpha glycosidic bond, what you simply mean is that you've got these two glucose units, right? These are monosaccharides. So you've got one glucose unity, another glucose unit. When you combine them, they form in alpha glycosidic bonds. Since we've said that the alpha glucose has got the OH group down, when you combine it with another glucose unit, it's going to form what we call an alpha glycosidic bond. Because why? The OH group is down. Even here the OH group is down. But why are we calling it 1,4? If you look at your first glucose unit, this OH group that you are getting here is at carbon atom number 1. If you go to the other glucose molecule, if you count from here, 1, 2, 3, find that it's at carbon atom number 4. And you get a hydrogen atom from here and this OH. You combine, remember, hydrogen plus OH form what we call water. 2 hydrogen atoms plus oxygen, they form what we call water. But as you do that, you form what you call an alpha 1,4 glycosidic bond. Your beta glycosidic bond is different. Why? It's because your OH group, remember I said the OH group on the beta glucose molecule is on top of a C1 carbon atom. So when you combine it with another, whether it's an alpha glucose, as long as the unit that you're combining it with is a beta glucose, it's going to give you a beta glycosidic bond. Why? When you combine this one, look at this. This is your C1. You've got your OH group on top. When you add another glucose molecule, even if you got this one, you add it here, it's going to give you a beta. Why? It's because on a beta glucose, you've got this OH group on top. Now it's forming this diagonal shape here. You get your OH group there with your one hydrogen atom that you get from there, then you form this 1,4 beta glycosidic bond. So these bonds will be very important, especially the alpha glycosidic bond. It's going to be important when I talk about the actual glycogen molecule. Alright. Now, one last recap. Look at what is happening here. So you've got what we call 1,4 and 1,6 alpha glycosidic bonds. So what you are looking at here, just that you've not noticed yet, of course you're going to learn, is primarily the structure of glycogen. But before I talk about that, I just want you to understand the difference between 1,4 and 1,6 alpha glycosidic bonds, of which I believe that you've already understood the 1,4. So 1,4 alpha glycosidic bonds is what I'm talking about in the previous slide. You've got these two glucose units here. There was an OH group down here, OH group down here. Since the OH group was down, it makes it alpha. When they combine, they form the alpha glycosidic linkage. But why are we learning about these things? The glycogen that we want to form is usually branched. It has got branches. So it has got this straight unit of glucose, but it has got a branch here. Now when you're forming that branch, you form what you call an alpha 1,6 glycosidic bond. Focus on this glucose unit. Let's say you've got a straight glucose unit of glycogen, right? Then you want to form a branch. When you come at this specific glucose molecule, if you count from here 1, okay, you count your carbon atoms from here 1, 2, 3, 4, okay, you've got 5 there, then this is your 6th one. So as you form the branch, as you add another glucose unit by forming the branch, you form it by attaching this glucose unit here at carbon atom number 1 with this glucose unit here at C6 or carbon 6 glucose. And you're going to form what you call alpha 1,6 glycosidic linkage. So I hope that is clear. Okay. Alright. So let's make progress. You must also understand that, remember in the introduction I said glycogenesis takes place in the liver and in the muscles. Now, if you are talking about glycogenesis in the liver, we are going to use an enzyme that we call glucokinase. If you watched my video on glycolysis, you remember this. When you convert your glucose to glucose 6-phosphate, If it's in the liver, you use what we call glucokinase. If it's in the muscle, you use what we call hexokinase. So I just want you to have this understanding. Just keep it for now. Any questions? I believe there are no questions. So now let's look at the actual pathway that involves the glycogenesis. Alright, sorry for that. So let's look at the actual pathway that involves the glycogenesis. So I'm going to talk about glycogenesis in the liver. Remember I said it can take place both in the liver and the muscles. But in this video, we're going to refer to the liver. So imagine, remember we said it's taking place in the fed state or when the glucose levels are above 7.80 millimoles per liter of blood. So imagine you are sitting on the dining table, right? Then you are eating your shima, or you are eating that rice, or you are eating whatsoever you are eating which has got carbohydrates. And then you start obtaining glucose, from that. So when your glucose levels are high now, when they are above 7.8 millimoles per liter of blood, you are going to initiate this process of what? Glycogenesis, which takes place in the liver, specifically the cytoplasm. So on your left side here, I'm just trying to show you or remind you to say you've got the excess glucose, this is, remember, just consider this as a blood vessel. Imagine it's a blood vessel, then I'm going to use this diagram here just to give you a reference point of a glucose molecule. This is your first carbon atom, fourth carbon atom, the 60th carbon atom. So let's assume you've got your excess glucose levels in your bloodstream here, from your blood vessel. So what are you going to do? You're going to transport, this glucose unit from the blood vessel to what? To the liver, because you want to store it. You want to convert it to glycogen. So imagine you've got a lot of them, and you get one of them, it goes into the liver. So there's a transporter that you find here as you transfer this glucose unit from the blood vessel to the liver. There's what we call a GLUT2 transporter that you find here on this membrane. Okay, so this is called GLUT2, glucose transporter type 2. If it's in the muscle, it's called glucose transporter type 4. So you've gotten this excess glucose unit from your blood. You've made it enter the liver cell, specifically the cytoplasm, where glycogenesis takes place. What happens first? So what's going to happen first to that glucose molecule that you've put inside the cytoplasm is that you're going to convert it to what we call glucose 6-phosphate. And you and I know very well to say the enzyme that converts glucose to glucose 6-phosphate in the liver, we call it glucokinase. But wait, why are we calling it glucose 6-phosphate? It's because in this particular stage, we are using an ATP molecule. That's why we are able to add the phosphate group at carbon atom number 6 of this glucose molecule. So, we've gotten our excess glucose from the blood, made it enter the liver. Then the first stage that we did was to convert it from glucose to glucose 6-phosphate using glucokinase enzyme, and also by adding a phosphate group at carbon atom number 6. And we're calling it glucose 6-phosphate. And we used an ATP molecule in this stage. Then let's continue from there. Now, what happens is that your body doesn't want this glucose 6-phosphate. It's like, no, I can't use this. What I want is a glucose 1-phosphate. Very simple. If you already have a glucose 6-phosphate and your body needs a glucose 1-phosphate, you just shift this phosphate group from carbon atom number 6 to carbon atom number 1, because the body wants glucose 1-phosphate. So that's what we're going to do here. We're just going to shift this phosphate group from C6 to C1 using what enzyme? Phosphoglucomutase enzyme, which converts glucose 6-phosphate to glucose 1-phosphate. Recap. You got your excess glucose from your bloodstream, made it enter the liver. In the first stage, you converted it from glucose to glucose 6-phosphate. From glucose 6-phosphate, your body doesn't want this. It wants a glucose 1-phosphate. So you're going to do that. You convert your glucose 6-phosphate to glucose 1-phosphate using a phosphoglucomutase. Then from there again, your body is like, again, I can't make use of the glucose 1-phosphate. What I want for me to form glycogen is what we call a UDP, which is connected to a glucose unit. Now, the body is like, okay, I've got the glucose 1-phosphate already. Then how do I make this UDP? Remember, UDP simply means uridine diphosphate, which is attached to a glucose unit here. So there's a solution already. In your cytoplasm, you've got what you call UTP, right? Uridine triphosphate. It's already there in your cytoplasm. So the body, what it's going to do is that since you've got UTP, remember UTP is uridine triphosphate, meaning you've got 3 phosphate groups. But this glucose unit that you have here, glucose 1-phosphate, it already has 1 phosphate already, meaning the body is only looking for 1 to make a UDP. So what it's going to do is that this UTP, it's going to cut off the 2 phosphate groups and just remain with 1. Then they fuse now. Okay? Remember, if you remove these 2 phosphate groups, it's no longer called UTP. It will be uridine monophosphate. Then this uridine monophosphate now, it's going to add with this glucose 1-phosphate and they form UDP. Why is it becoming diphosphate? Because you already had the phosphate group here plus this 1-phosphate group that you've gotten from this UTP. When you add them, you form uridine diphosphate that is linked to this glucose unit. That's what happens up to this stage. Okay, so let's proceed. We've converted our glucose 1-phosphate to a UDP glucose that is linked to a UDP glucose. Uridine diphosphate linked to a glucose unit. Now, there's something that is special again in the cytoplasm. So in the cytoplasm, you've got what you call glycogen. It's a protein molecule that has got an OH group which attracts these UDP molecules that you formed. So this glycogen now, it is the one that now start attaching itself to these UDP glucose molecules, and as it does that, it starts to form the glycogen molecule. So you've got a glycogen in your cytoplasm already, and if it sees to say, okay, there's a UDP there. Remember I said this glycogen has got an OH group which attracts these glucose molecules. So what will start happening now? Remember, if you've got excess glucose, they are coming from your bloodstream, okay? They enter the liver. They are converted to glucose 6-phosphate, converted to glucose 1-phosphate, converted to UDP here. Then they'll start now being attached to the glycogen. So it attaches another one, another one, another one, another one, until you form this long chain of a glycogen molecule. But you must understand the following things. Remember, this glycogen is already there in the cytoplasm. As you keep on adding these, glucose units here, remember at this stage you've got a UDP. You've got this glucose unit that is attached to a UDP. But as you add this glucose unit on this glycogen, you are removing the UDP. It's being cut off on its own. There is no enzyme that is involved here. There's what we call autoglycosylation process. Okay? So what you're just doing is that when your body forms your UDP, this UDP, you get it, you just remove this glucose unit, you attach it to a glycogen. You get another one, you attach it, you get another one, you attach it, and you'll be getting rid of these UDP molecules. These will be used later. They're going to combine with these phosphate groups that were cut off here to form another UTP, which another glucose molecule can use again in the same process. So just keep on adding this glucose unit on this glycogen and elongating the chain. Now, if you form about, from this glycogen molecule, if you add about 7 to 8 glucose units, those 8 that you've added or 7 that you've added do not need an enzyme. They use a process that we call autoglycosylation. We're calling it auto because it's self-regulated. It doesn't need any enzyme. It happens on its own. Now, just assume you've got this glycogen. Let's assume we've made about 7. I just didn't draw 7 because of space. Let's assume we've made about 7 of them. So there is what we call a glycogen synthase enzyme in the cytoplasm. It is the one now that elongates this chain beyond the 7 that you had formed initially. It's going to make it so long, depending on the glucose levels that are coming from your bloodstream. Okay? It's going to make it so long. So if this glycogen synthase now, if it notices the pattern to say, but why is this chain elongating? I think we're supposed to form glycogen. So if you form about 7 of them, then it's going to continue the journey from there. It will keep on adding this glucose unit on top, until it makes a long chain. But what I also want you to understand that this straight chain is made up of what we call alpha 1,4 glycosidic bonds. Those that I've shown you in the previous slides. you can see that your C6 carbon atom number 6 is on this side, meaning your 1 and 4 are in between here. They are the ones now that are making these linkages. Alpha 1, 4 glycosidic bond. They can ask you this question. The straight long chain of glycogen, the straight one, is made up of what you call alpha 1, 4 glycosidic bonds. But you know, your body has got common sense. It knows to say, if I keep on elongating this chain, I might reduce the surface area. Because what if I've got a lot of glucose which are coming here and I must form a lot of glycogen? But if I keep elongating this chain, I might reduce the surface area. So it will start branching. It will start forming branches now, just to make it look like a tree. So how does it form those branches? It uses what we call a branching enzyme. It's already there in the cytoplasm. Okay, a branching enzyme. So what it's going to do, let's assume we've got this long chain up to here, then this branching enzyme is going to cut it from here. Cut. If it cuts it, it gets this piece it has cut and branches it on this end here. Now, each branch that is formed in a glycogen is made up of what we call alpha 1, 6 glycosidic bond. I had shown you how this bond is formed. I said this bond is formed when you are forming branches. Remember, a glycogen molecule has got branches. So, if it's the straight line or the straight unit of glucose, you just have 1, 4 glycosidic bond, alpha. If you start forming branches, at that particular branch, when you attach another glucose unit, you're going to form what we call an alpha 1, 6 glycosidic linkage. What you are seeing on the end here just now is showing you the long glycogen molecule. This is the glycogenin, then this is the straight line of glucose which has got 1, 4 glycosidic bonds. But as you form these branches, at every branch there is what we call alpha 1, 6 glycosidic linkage. Summary of this process that I was talking about. You've got a glucose unit, remember, in your liver, okay, cytoplasm specifically. Your glucose unit, if it's in the liver, you use glucokinase. If it's in the muscle, exokinase. You convert your glucose to glucose 6-phosphate. But your body doesn't want this glucose 6-phosphate. You convert it to what we call glucose 1-phosphate. What do we use? Phosphoglucomutase enzyme. And then I said your body again doesn't need this glucose 1-phosphate. It wants a UDP glucose. That's why you utilize that UTP that I had said. Okay? You remove the 2 phosphate groups, remain with 1. Then you've got another phosphate group here, meaning when you combine them, you're going to form a UDP. Okay? From there, you're going to start adding now this UDP glucose to glycogen that I was talking about. And you're going to have autoglycosylation when you add about 7 of these on this glycogenin. Beyond that, if they are beyond 7 or 8, you start using glycogen synthase, which now elongates the chain of this glycogen, depending on the number of glucose, molecules that are coming from the bloodstream. Summary here. We are saying that the glycogenesis summary, look at what is happening here. You've got this long chain of glycogen. Okay? So this straight long chain, okay, is being elongated by what we call glycogen synthase. Alright? Glycogen synthase. If you count these in green, there are about 1, 2, 3, 4, 5, 6. Let's say about 7 of them. And I said this takes place using autoglycosylation. But beyond that, going up, this glycogen synthase now is going to say, I think we're supposed to form glycogen. It will keep elongating this chain. But if this chain becomes too long, it will start forming what we call branches, using the branching enzyme. And I said at every branch you've got an alpha 1, 6 glycosidic bond. In the straight line or the straight unit chain, you've got 1, 4 glycosidic bonds. That's all about glycogen synthesis. Let's talk about the regression, which is the last thing we're going to talk about. I said earlier to say insulin is going to promote glycogenesis. That's why you always give, insulin to diabetic patients so that you can reduce the number of glucose in the blood. What do you just do? You are just promoting the process of glycogenesis by giving them that insulin so that you reduce the number of glucose levels in the bloodstream. Glucagon, it does the opposite of what? Glycogenesis. Glucagon, remember, is a hormone that breaks down the stored glycogen back to glucose. But you ask yourself to say, what you want to do is, we want to form this glucose. So meaning this is going in our opposite direction. No wonder it inhibits glycogenesis. Talking about the enzymes, I've already told you to say, if you are talking about the liver, then there is involvement of what you call glucokinase, which convert glucose to glucose Then glycogen synthesis, I said here it's clear, after the autoglycosylation has taken place, it will keep elongating this chain now of glycogen by adding the glucose units on top, until it forms a long chain. Branching enzyme, I said this is the enzyme now that just try to make sure that it uses the surface area of the cytoplasm very well by branching this. It's going to cut here, gets this part and branches it here and keep elongating. So the one that elongates the straight chain is the glycogen synthesis. The one that forms the branches is what we call branching enzyme. I repeat, on the branch you've got alpha 1, 6 glycosidic bond. In the straight chain you've got what we call alpha 1, 4 glycosidic bonds. That's all that you need to know about glycogen synthesis, or what we call glycogenesis. What you're just simply doing is that you're converting that excess glucose into glycogen so that it can be stored. You're just making the stored form of energy so that later on again you can use it. In my next video, I'm going to talk about the opposite, which is glycogenolysis, where you're now breaking down that stored glycogen back to glucose depending on the needs of the body. Thank you so much. Make sure that you like this video and you subscribe to this channel.